back, everybody. In this video lecture, we're going to be exploring our very first specific topic of the class. And what we're going to be doing in this particular topic is examining how our biological mechanisms form and change throughout the lifespan. In this specific lecture, what we're going to be doing is really starting where it all started. In particular, we're going to go back to when we were all just one tiny cell. In fact, many of you probably already know this, but life begins for all of us as one tiny microscopic cell that starts to multiply and replicate like crazy. What are the details of this singular cell? How can we study something like this in its even most basic form? Well, first of all, when we're talking about this single cell, we should probably give it a name. The name given to it by scientists is something called a zygote. It's the first cell that all of us are as we start to multiply and become the individual we become. Within that single cell are the critical elements required for the cell to not only replicate, but eventually all the different structures within us to start to form. And the main catalyst for this information was this collection of tube-like structures found within the nucleus, the center of all these cells. These structures are called chromosomes. Each cell within our body, at least most of the cells within our bodies, and definitely all of our zygotes, have 23 pairs of chromosomes within them. There's always some anomalies that we'll talk about a little bit later in the next lecture, but for the most part, all of us start off as a single cell with 23 pairs of chromosomes found within it. Within those chromosomes is all the information required for us to start to grow. In particular, when we're talking about growth at this level, what we're talking about is finding a way to get that single zygote to multiply into 2 cells, and 4 cells, and 8 cells, and so on, until we're billions and billions of cells within our body. This process of multiplication is something called mitosis. It's kind of a 4-step process that occurs over and over again throughout our lifespan. And to understand how mitosis works and its importance, I thought I'd give you kind of a second voice on the matter. It's a video I found a few years back that I found extremely interesting and somewhat humorous. Varies from person to person as to how humorous they find it. But you'll see on the class lecture notes a link to this video that covers the process of mitosis. I encourage you to watch that now and then come back after you've learned about how these cells can replicate through this process. And we'll keep going on to other things that happen as these cells start to reproduce and the sources behind that reproduction. If you've watched that video, you've seen some really cool, interesting information on how this unfolding process of mitosis actually happens. I also talked about the utility of it a lot in the video. But one of the questions that I noticed most often isn't answered in videos like that and other places is, how do the cells know to replicate in the first place? Well, this brings us back to the strand-like structures that we talked about earlier called chromosomes. As we talked about, within our nucleus, we have 23 pairs of chromosomes. And within those chromosomes, if we splice them out and really look at them closely, kind of untangle the mess that creates the strands, we actually get to see this double helix-shaped information that we call deoxyribonucleic acid. The short is just DNA. And this complex material is actually the code to all of the cells in our body, code for all of the cells in our body. That gives it a lot of information. Partially, it tells these things how to reproduce themselves, but it also tells them lots of additional information. And that additional information is found within the information on the DNA that we have. This is these complex proteins that we call genes. Genes come in abundance within each and every cell. And genes, we think, are really the catalyst behind the process of mitosis, but also the catalyst behind a lot of other things that occur within our cells that we kind of take for granted. As we start to develop, Genes do more than just randomly cause our cells to reproduce. One of the things that they do as we start to become more complex in that multiplication process, as we transition from a zygote to a couple hundred cells, is they start to dictate what types of cells this multiplication process is going to create. We'll learn in our next class about there being 3 different types of cells that start to form in the early phases of development. By the time we become humans, or humans, but by the time we're born, I guess, there is a lot more complexity to the cells than just having 3 types of cells within our body. And the DNA contained within all of our cells is a big player in not only the types of cells that are formed, but how they form at different points in our life. As we start to actually grow and more cells are created, ones that are more differentiated, the DNA within our cells also tells these cells to move into specific spots. That actually creates proteins that tell these cells to move into different spots. And this is really a big component to the development of a human being. It's a big component to the development of all species. We not only need lots of cells, but we need them to be in the right place and to form at the right time for correct development to occur. Another thing that happens as these cells do start to move into places is they move there to create the physical structures of our body. And this not only includes our physical body, but a lot of the internal things that are really critical for us to be able to survive. Our organs have to be formed at specific times, our nervous system and all the different things happening at the microscopic level have to be formed. And all of this is really taking place as a byproduct of the messages that our DNA is sending, or I guess the genes within our DNA is sending to all the cells as they start to grow and move in different directions. And this particular effect continues throughout our lifespan. Not only does this DNA help us form our body when we're born, but it also continues to help our body to grow, change, and if we're looking at the nervous system and other structures of our body, it also dictates how our body reacts to the environment around us as we start to get exposed to different things and different elements that give us that unique characteristic of who we actually are. So in essence, even though we talk about genes and DNA as being key players in our cells replicating, that's just a small portion of the story behind how our genes really allow us to become who we've become. And this idea of genes being so unbelievably powerful is something that led many scientists in the late 1900s to become enamored with them. When we started to realize that genes were the building blocks, all the different things that made us who we were, many scientists around the world flocked to this notion that if we could break that code, if we could understand everything that was in that DNA that we had, we could maybe understand a lot more about human nature. And it led to this endeavor that we nowadays call the Human Genome Project. It officially began in about 1990 and lasted for about 13 years, where scientists all over the world were trying to figure out how to sequence the genes of individuals, of human beings in particular. We'd already been able to sequence the genes of some less complex organisms in the world, but humans were extremely complex compared to a lot of them, and scientists were trying to find ways to figure out how to code these proteins and make sense of it so we could theoretically break the puzzle of human beings. And eventually, in 2003, we did have success in that we were able to sequence the human genome. In fact, the first genome that was sequenced was a genome of one of the lead scientists on the particular topic. And if I recall correctly, the particular gene sequence that was sequenced was put on, I think it was Time magazine, the exact code of this individual. And it led to other people sequencing other genomes and eventually got us to where we're at today, where pretty much anybody going into a doctor's office can get their blood drawn and have their entire genome read off to them. I don't know if you'd want it read off to you. It's 20,500 genes, but you could have that information on display and you could use it for whatever random purposes you might want to nowadays. And it has had some utility. What we've been able to do with the sequencing of the genome is identify specific genes and gene clusters that are linked to specific characteristics, be they mental, physical, emotional, lots of different kind of areas that we've gone into. So once we've been able to sequence the genome and really start to link specific gene characteristics to individuals, I do want to caution you when you start to kind of become enamored with this endeavor of the human genome that even though scientists saw this as the holy grail and kind of the finish line to understanding large amounts of the human process, what we've been able to find is that actually this human genome that we're sequencing is only telling us part of the story. What we've discovered lots of times is things that we linked specifically to genes forever, or just assumed were linked to genes forever, are much less clearly linked to some of these experiences that we have. In essence, there's this need for the environment to kind of happen, not only at specific points, but in specific ways for genes to create lots of things to emerge. And there's not just the occasional back and forth going on between genes and environment. It's a continual orchestra that's happening all the time at different stages in our lives, where people with the exact same genetic information might come out of a situation with very different reactions or very different byproducts of that event, simply because of the way the genes of that individual interacted with that situation. Subtle, tiny things can have big, big effects. And this realization led lots of people away from this belief, this notion that we could essentially understand everything about you just by sequencing your genome. We now believe that you really do need to know not only a person's genome, but all the different little microscopic life events that are shaping the lives of these individuals to understand them fully. Now, some of you might be asking, now that we've covered where these genes are coming from and how it tells our cells to multiply, well, how was the zygote created in the first place then? What led to that creation of the very first cell? I am sure at some point in your life, your parents or somebody else that you've talked to has told you about things like the birds and the bees and how sexual reproduction happens. But at the cellular level, there is a lot of really interesting information to explore. When we talk about a zygote, what we're really talking about is the very first cell that's come from two what we sometimes call parent cells, or what we sometimes call gametes. And for human beings, the two gametes that need to come together are a sperm cell and an egg cell. Gametes are very specialized cells that actually are kind of unique in their design and their chromosome composition. Sometimes we call gametes haploid cells, cells where we don't have 23 pairs of chromosomes within them, but just 23 chromosomes, ones that are kind of unique in their own nature. For most of us, when we look at the cells within our bodies, most of them are these typical cells with 23 pairs of chromosomes, and they all contain the exact same chromosomes. In special areas of our body, we do have these haploid cells that are all very unique. And the way they become unique is through this really interesting process that's called meiosis. And again, to give you some clarity on this very complex process and also provide some brevity in the process, I thought I'd give you another video link that is from the same creator of the last video that you saw that goes over how meiosis happens, what makes it unique, and also the byproduct of meiosis. So again, if you can, pause this video, take a step out, and watch the other video that covers the really interesting process of meiosis. We'll come back after that. If you've watched that video, hopefully you've gotten some really interesting information on how this meiosis process actually works. And you probably have also noticed that even if we just look at meiosis, there's more complexity to how these single cells, these haploid cells, are actually formed. one of the key players in the development of these individual cells that provides a lot of individual variety is this thing that was briefly mentioned in the video called crossing over. It's this interesting step that occurs when two chromosomes actually line up next to each other, overlap, and exchange genetic information. Now, this might seem like kind of a random effect, but what it does through this crossing over process is ensure that essentially we've got variety in the cells, and that any issues, any abnormalities within the cells can sometimes be overcome. And crossing over tends to allow the healthier genetic information to kind of thrive, and the less healthy information to sometimes fall off. This is something that's extremely noticeable in research that's recently suggested that the older we get, the less crossing we have through the cells that are formed through meiosis, and more likely we are to have lots of genetic abnormalities and other problems that we'll be talking about in the next lecture. So even though crossing over, this weird overlapping of cells, might seem like a random nonsense thing that we're just throwing in there at the last minute, I want you to recognize that there is a lot of utility to this. If you got to watch the video again to kind of catch that point where they discuss it, please do so. Also, if you want to read about crossing over, the textbook hasn't changed since this recording, you should find a little bit of information about it there as well. It's a really interesting process that's constantly taking place in addition to that meiosis process that's taking place and producing those haploid cells that we talked about earlier, the gametes of the egg and the sperm. But even with all of that in mind, we do always like to go back when we're talking about the splitting process and the creation of the zygote to what the byproduct is. And to understand this byproduct, I always like to have us do a little activity. Now, this activity is something that most of us can participate in and kind of go to the end, but I don't want to make anybody upset or offend anybody if we can't do some of the research that you're asked to do for this. Just bear with me as much as you can, and hopefully we can get something out of this. Because what I want you to do in this activity is think of some things that really define who you are right now. I think of 3 physical traits that you possess. It could be your height, it could be your speed, it could be your hand-eye coordination, it could be your flexibility, it could be strengths, weaknesses, neutral stuff. It doesn't really matter what they are. I just want you to think of 3 really distinguishable physical characteristics about you. And then after that, I want you to think of 3 things that really define you in terms of your personality. It could be that you're humorous or outgoing or sensitive or whatever. Think of those 3 personality traits, and then after you've done that, think of 3 cognitive characteristics that you link to yourself. Maybe it's you're somebody that's easily distractible, or you're somebody that loves to kind of explore creative stuff, or you're somebody that's really good at math, or somebody that has a preference for reading. Whatever it is, think of things that kind of define your mind. And after you've done this, I want you to see if you can trace those characteristics that you have back to certain individuals in your life. In particular, the producer of the sperm that made you who you were, the producer of the eggs that made you who you were, maybe the combination of them, or neither individual. If you can't do this because you don't know these individuals, totally fine. I'm not trying to make anybody upset with this. But what I want us to do with this activity is try to find a way to kind of link these weird findings that most of us will encounter with an issue that people run into when we talk about how these cells eventually form who we are. In particular, what I want us to recognize through this activity is that the formation of who we are is more than just kind of getting something from mom or dad or the egg provider or the sperm provider. It's usually this kind of interesting combination of effects that occur. In the past, we thought that you would get your eyes from your dad, your hair from your mom, and all of these kind of odd things that just were a direct byproduct of one parent. Nowadays, we've recognized through lots of genetic research that really what we're looking at when we see somebody's physical manifestation of something or mental manifestation of something is that thing, if we're linking it back to genes, is really a byproduct of what we call a person's genotype. This genotype is kind of the combination of genetic information that's coming from both haploid cells of the sperm and the egg. And it produces this thing called a phenotype. So in essence, your height, your eye color, all of these things are coming from both your mom and your dad, even if your eyes match your mom's or your hair matches your dad's. It's really both parents that are contributing to the emergence of that. And to make this even more complex, I want us to understand that phenotypes are very rarely in human beings the byproduct of just two cells coming together. Usually, when we look at phenotypes, we're seeing something that's a byproduct of multiple gene pairs that produce things. Something that is as simple as hair color is actually the byproduct of many different genetic combinations. Same thing goes with, I believe, even eye color and other elements to who we are. Right? We think of these things as being straightforward, but there's actually a lot of stuff going into it. Moreover, we don't just have then this basic formula that you see below that most scientists and biologists have used for years, one dominant and one recessive allele in every different genotype. Actually, often various genes that can be linked to specific areas on chromosomes. And sometimes you have what's called co-dominant alleles, co-dominant genes that can create interesting kind of byproducts when they come together. And all of this, again, makes the ability to predict who you are and what you're like as a result of your genes all the more challenging. Going back to that activity that we did, what it means is that even though you might have similar eyes or a similar propensity for word problems as one parent, doesn't mean that they're the ones that gave you the genetic information that produced that. Really, those things are a byproduct of not only both parents, but really a combination of genes that both parents provided you that led to the emergence of those specific characteristics. That's, of course, completely dismissing the importance of the environment as well. But even if we don't consider environment, what I want you to really get from that is this is a very complex process. And if we're looking at what makes it even more complex, as we talked about before, you have what's called polygenetic inheritance. We didn't give it a name until just now, but we have this thing called polygenetic inheritance, this kind of emergence of traits based on the combination of multiple genes. And we also have this thing that we mentioned earlier, where we have this heredity with environment interactions. So you have different genes being turned on and turned off at certain points in your life, and them being affected when they're turned on and turned off by the environment in different ways. This has led lots of researchers to focus their attention when talking about physical development, both inside the womb and long after, about this thing called the epigenetic view of development. This kind of recognition that when we want to understand who you are, we do need to know your genes, we do need to know your environment, but we also have to understand how that process of the interaction between genes and environment unfolded as you yourself unfolded. It creates a lot of interesting bumps in the road that creates a wide range of individuals that have lots of unique characteristics with them. And it does create kind of a mountainous task for developmental psychologists if they want to understand how you're becoming the person you become. In essence, going back to the analogy from our last class, if we want to really understand that snowflake, we've got a lot of things that we have to take into account. One other thing that's kind of worth noting here is that even with those things that are set, there can be some unique things that pop up in individuals that create unforeseen issues. There's this really weird effect called mutations that can happen because of exposures to viruses or other things, pathogens in the environment that can actually slightly alter our DNA. And that on its own is something that lots of scientists have started to explore over the last couple of years. Should probably note, though, that when I'm talking about mutations, we're not going into X-Men territory or any type of superhero territory. Mutations are really just tiny microscopic changes to our genetics that have really piqued the interest of researchers over the last couple of years and given us another wrinkle to throw into this complex story. Now that we've covered this topic of how we become the person we are and all these kind of interesting things that the kind of are the catalyst of the early formation of us, we're going to spend some time now looking at actual physical development. We're going to discuss how we go from that single cell into a newborn, and we're going to be doing this by focusing our attention, especially in the second part of this lecture, on a lot of the kind of nervous system things, the kind of brain and other characteristics that are linked to the brain that allow the mind to form as we're physically developing. We'll also spend a little bit of time talking about some things that can disrupt this path of development, just so we can have a more holistic view of not only the arc that we go through, but what happens when that arc is thrown off. But for now, thanks for listening. Again, I need to really stress, you want to watch those videos that I gave you links to, because they will provide some really valuable information. And when you're done with that, well, it's time to move on to the next lecture. You all have a good time, and take care.